I'm going to talk now about cellular immunity. And cellular immunity is the side of the immune response which is capable of recognizing and destroying our own host cells which have been infected by viruses, for example. And it's an ingenious system because technically you might think that once a virus gets inside one of our cells and it is surrounded by our own cell membranes in our cells, that it will become invisible. And on the face of it, this would be the case. But there's an ingenious system that we have with cellular immunity where cytotoxic T cells or natural killer cells are able to kill cells that have been infected. And we're going to learn about how that happens now. The first thing to say is I've got the schematic up here, which I used for human immunity, which takes place on the left-hand side, really, involving B cells. But it's a very similar process in its nature in order to get the activation of cytotoxic T cells. Because remember, we're dealing with a pathogen here which has a certain antigenic variation, so a sequence of polypeptides or glycoproteins, glycolipids or polysaccharides. And that pathogen will have been recognized by the antigen-presenting cells, like a dendritic cell or a macrophage, in the same way as the humoral and the cellular. And that will have led to an activation of a T helper cell. And the T helper cell could, as we saw with humoral immunity, activate a B cell. But it's also able to activate cytotoxic T cells by releasing cytokines. And the cytotoxic T cells that bear, and this is crucial, that bear the correct T cell receptor for that antigen will be stimulated to replicate, and loads of mitosis going on, and we have proliferation of the right cytotoxic T cell for that antigen. There's one big difference, though, about the T cell receptor on the cytotoxic T cell. And that is, rather than docking and being complementary to MHC2 carrying antigen, which T helper cells are, it is actually complementary to MHC1 holding the antigen. And this is crucial because our normal body cells express MHC1 proteins on their surface, on their cell membranes. And that means that that's the docking MHC that we need to be dealing with here, not MHC2. So that's a fundamental difference between cellular immunity and humoral immunity. And so how does this work? Well, if we go to a video here, I'm going to see if I can get it to the right place for us. If we pause this here, we can see that we have a virus-infected cell here, and in the background is a cytotoxic T cell. And hopefully it should come back, great. And the cytotoxic T cell has got a T cell receptor, which we're about to see, but it's very similar in its nature to the ones we've met earlier. And that T cell receptor is going to be complementary, as I say, to MHC1 holding antigen. And so what's going to happen here is we know from our biology before what's going on in this virus-infected cell. Inside this cell, we have got, right now, if it's been infected, we know that the virus injected its DNA or its RNA, which went to the nucleus. And we know that the DNA, for example, would have been replicated. Okay, so we've got viral DNA, which would have come out into the cytoplasm. At the same time, we would have had protein synthesis. So we would have had messenger RNA produced, which went out into the cytoplasm and was formed, ribosomes formed viral proteins. And then we know that these proteins and DNA, for example, would have assembled, and normally they would have gone out of the cell, causing lysis, and we would have had maybe millions, hundreds of thousands of new viral particles, which would then go on and infect other cells. So our normal viral pathway is injection, replication of DNA, protein synthesis, packaging them back up together in the cytoplasm, and then lysis. Now, ordinarily, that puts the cell in some difficulty. But the ingenious nature of this immune response is that this cell, which has been infected, is capable of telling the rest of the immune system that there's something not right going on. And so what it will do is this cell will take these viral proteins which are being made and recognize them as non-self. And it will then, using endoplasmic reticulum and Golgi apparatus, package them up, put them into vesicles with MHC1, and the vesicles will exocytose and express MHC1 plus the antigen. And so what this body cell has done, just a normal body cell, is effectively said, everyone, I don't like the look of this. There's not me, there's not self in being made inside me, and here it is. Is anybody interested in that? In the same way that antigen presenting cells, like macrophages and dendritic cells do at the beginning. And this antigen with MHC1 is, as I've mentioned, complementary to the T cell receptor of the activated cytotoxic T cells, which have been activated by the T helper cells earlier. And so we're going to watch now what happens. So we're going to see what happens when this T cell with a T cell receptor is able to locate and dock with the MHC1 and cause lysis of this cell, the death of this cell, killing everything in it, but importantly, killing the viral particles before they've had a chance to assemble. Okay? And I'm going to talk you through it. It's incredible. The last thing to say is this: as well as causing the death of this cell, we're going to have other cells around here which are very similar that may or may not be affected. And what the cytotoxic T cell is going to do is it's going to release chemicals which actually will be accepted by specific receptors on these neighbouring cells, which cause cell death in them as well. It's like a belt and braces approach, making sure that that cell's definitely infected, but you know what? Maybe other cells have been infected. And so you'll get surrounding cells being destroyed as well, just to make sure that there's no or as much less chance of infection. Okay, so let's have a look at this video, just moving forward, and see how this works. So here's our virus infected cell, and here's a toxin. Now, I love this video because it's got the tertiary structures involved, and you can see the viral antigen there being held by MHC1, and we now have this reaction due to the complementary T cell infector receptor, and we've got activation of the killer T cell. This releases cytokines, and this now is going to do something. It's going to release perforin, which is a protein which assembles on the infected, so this is the viral infected cell, it assembles and it creates a doorway for a hydrolytic destructive enzyme to enter. And so here comes the granzyme, and so we have created a door, and the granzyme hydrolytically digests the cell, killing all of the viral proteins and DNA at the same time. So I think that's beautifully elegant. We have a cytotoxic T cell, which will dock into MHC1 plus antigen, and when it docks, that will cause an activation of that cytotoxic T cell, and that will release cytokines, which then cause perforin, a protein which forms a channel in the cell membrane of the infected cell, which allows granzyme, a hydrolytic digestive enzyme, which digests the cell, and that causes cell death of our own host cell, which has been infected. And as I said, surrounding cells would also be effectively asked to fall on their swords and die, which is called apoptosis. in order to make sure that there is no further infection in the surrounding areas. And so that is how we deal with our own cells being infected by viruses.